Alright, I'm going to take you guys through how to identify cell types and tissues when looking at a transverse section of a leaf. Something you should be familiar with for the prac assessment and also the cells, and probably even later the transport and plants topic. So just talking about what a transverse section is. In a root or a shoot, a transverse section would be like this, usually cut down the side of the cylinder. A cross section is like cutting the end of a pipe off, and then looking at the circle at the face that you've just cut through. In a leaf, a cross section and a trans section are often used actually to refer to the same thing. So you can see the picture here. You're essentially cutting across the leaf and looking at the face, the surface that you've just cut. Now, when preparing these slides, they actually have to cut a very thin slice out of the leaf, because if we were trying to look at that cut surface, obviously it would still be very, very thick. So let's get straight into it. Let's look at some of the tissues. So here we've got a leaf cross section or a transverse section through the leaf. It's actually a little bit different because we've punched a square out of the leaf, and this is what we're looking at here. The transverse section or the cross section would be this face. So normally we wouldn't see this. Normally we'd see this face, and it'd be a very thin layer, hopefully just one or two cell layers thick. But we'd be looking at the side here. So going on, the first thing that you'll see, or sometimes see, is on the surface of the leaf, and sometimes it can be on both surfaces, but often more pronounced on the upper surface, is the cuticle. Now the cuticle isn't actually a type of cell or anything like that at all. The cuticle is actually a secretion from the epidermis, and it's just a waxy layer that prevents water being lost by evaporation. Now sometimes you'll see a cuticle, sometimes you won't see one at all. It might be way too thin, or it might not be there, may barely have any cuticle at all. And sometimes it can be quite a thick layer, like equally thick as the epidermis itself, or even thicker. It can be on both sides of the leaf, but is often more pronounced on the upper surface. And it just prevents water loss, water being so crucial to the process of photosynthesis. It's one of the reactants in that process. So not enough water or losing too much water would prevent the efficient production of food, or store of energy through photosynthesis. And also, I mean, a plant's trying to maximize its light absorption by increasing the surface area, so it has all these leaves with a large surface area, but having such a large surface area does make it prone to water loss, especially considering it's exposed to so much light. So this is one way to try and minimize or try to balance that increasing light exposure and absorption while minimizing water loss. Cool, the next one is the epidermis. This is a protective layer. It's on both sides. You've got an upper and a lower epidermis. And these are cells that essentially act to protect the leaf, and they are responsible for producing the cuticle. So as a tissue, we could consider the epidermis and the cuticle as a tissue. Remember, the definition of tissue is cells of the same or a different type working towards the same function, and it includes their secretions. So, all right, next, down the bottom we've got these stomata. Stomata are the little pores. Now, technically it is just the hole, but in a lot of books and things like that, or often people will actually use the term stomata to refer to the entire structure, so the cells and the hole. Technically it is just the hole in the middle. The two cells that make up the stomata are called guard cells. And they can change shape slightly just to open and close that pore. And again, their job is to minimize water loss. So a plant, to carry out photosynthesis, needs to let in and out gases like carbon dioxide and oxygen. So it needs carbon dioxide to come in and needs to allow oxygen to diffuse out. But when it does so, water can also evaporate and leave in a gaseous form through this hole. So if it's very hot, the conditions are very hot, and the light intensity is very strong, a plant may actually have to minimize or reduce the amount of photosynthesis it can carry out, reduce the amount of carbon dioxide and oxygen that can pass through here, in order to prevent losing too much water. So these can help regulate the flow of gases in and out, to again balance, trying to maximize carbon dioxide intake and oxygen release, again being reactants and products of that reaction, so being very important, with water loss, so very important to make sure that not too much water is lost. Some plants have a very special ability to basically carry out some of the first steps of photosynthesis and store the carbon dioxide that's been produced, then open these stomata during the night when they're not going to lose so much water and release the carbon dioxide then. It's a good strategy in really hot conditions, so like desert climates, but it does limit the overall ability to carry out photosynthesis. So for plants that aren't in quite so hot climates, would tend to open their stomata during the day, at least partially or part of the day, and if it gets too hot, they close it up a little bit. So remember, these two cells are called guard cells. Now, in order for gas to flow in and out effectively, we need some air spaces in there. We'll talk about those in a minute. So these cells up here are called palisade mesophyll. Now, mesophyll literally means like middle leaf. Meso means little, middle, and fill comes from means leaf. So I think that stems from the Latin. Palisade, if you think of the word palisade, is a type of column. Right, so think of the big Roman sort of type buildings, and that's partly where they get their name from. They tend to look like, have that sort of column shape to them. So the palisade mesophyll are essentially a layer that are predominantly, their function is simply to carry out photosynthesis. That's where you'd find the highest number of sort of chloroplasts. So their job is basically to carry out photosynthesis. Underneath that, we've got the spongy mesophyll. So the spongy mesophyll is essentially also contains a lot of chloroplasts, but here the characteristic thing is they're not so column shaped, and the name spongy, just like a sponge, it has lots of air spaces or gaps in it, and this is where that carbon dioxide can move in and oxygen can flow out from, so to help exchange those gases in order so that photosynthesis can continue. Alright, now we've got the vein, or sometimes also referred to as the vascular bundle. This is basically for the transport of water and food. So here we go. Now this diagram isn't the greatest because it doesn't always have this sort of structure, but the phloem tend to be smaller, and often it's very difficult to see the individual phloem. They are often much smaller. The xylem tend to be much larger cells, and they often look very hollow, and indeed they basically are pretty hollow. They're just full of water. The cells of the xylem are generally dead, and so it's just essentially their cell wall, or that outer layer that remains, that acts as a pipe, and the inner cell is essentially just empty, and it's just there to transport water. So the xylem are generally larger, but they're not always arranged like you see here. So sometimes you might have one on top of the other and surrounded by what we call bundle sheath cells, which just help to prevent water and things leaking out from there. So the bundle sheath cells often, this diagram, if you saw this under the microscope, you might assume these ones around the outside are bundle sheath cells. But we'll look at a few actual examples. The main thing is to note you'll see them in a little bundle, and your phloem are usually smaller than your xylem. They're pretty easy generally to pick out which are your phloem and your xylem. Alright, so those are the main parts of the leaf. I'm going to talk more about so a lot of these are actually just cell types or cell layers. Talk a little bit more about tissues. So the palisade mesophyll and the spongy mesophyll, and even technically the epidermis, make up what we call the parenchyma. Now the parenchyma is essentially the majority of cells in most of the organs of a plant. Parenchyma actually exist in animals. In animals, the parenchyma are the functional cells, whereas the stoma in animals are the structural ones. So normally we talk about an organ having functional cells in it, and then structural, which are the connective tissues which hold the organ in place. Like at the end of muscles, you've got tendons which hold them in place, are the connective tissues. So parenchyma is kind of the functional filling tissues. So think, parenchyma, the functional filler tissues, and most tissues are actually going to be parenchyma. Now sclerenchyma is another type of tissue, and that's generally the bundle sheath cells that surround the xylem and phloem. And xylem and phloem technically can be considered their own tissues too, because they have a different function. There's another type of tissue, collenchyma, and you can't really see that on this diagram. So I'm going to shift to a transverse section that was taken under a microscope. So here we go. So this is a real image of an actual leaf. So let's try and identify some of the tissues here. So at the top we've got the epidermis. I can't make out a cuticle. All right, probably just too thin to see, or maybe it has a very thin or barely any cuticle. On the lower surface we've got an epidermis there too. Alright, now out here on the sides we can see the palisade mesophyll, nice and sort of rod shaped, spongy mesophyll with lots of gaps, and then in the middle here it gets a little bit more complicated. So in the middle I can make out this kind of circle. So this is the central vein, alright, and it consists of xylem. I can see bigger circles in the top, the upper part, and phloem I can't really see circles, it's much more sort of dense. Those would be the phloem down the bottom here. Now there may be sort of bundle sheath cells around the outside, a little bit difficult to see. If you can't see them so clearly, just draw it as a circle and label, xylem as the larger circles and phloem as the smaller, tighter ones. Now, here's where we actually get some collenchyma. So at the very top and at the very bottom, we get some collenchyma, just underneath the epidermis at the top and the bottom. Now the best way I can describe collenchyma is their job is often to provide extra structural support. And you can see that not very clearly in this picture, but they often have, or one definition of collenchyma is that they have thickening of the cell walls. They have slightly thicker cell walls. And often it's asymmetrical, so they're thickened in one direction or on certain sides, and that's to help provide structural support. And that makes a lot of sense that we have some of those at the top here and at the bottom here. Because if you think of the leaf like a plane, one of the key stress points where there's a lot of force acting on a plane is where the wings attach onto the fuselage, onto the body. And you'll know that if you've ever flown like model planes and things like that, when they break, it's often the wing that snaps off, and when it snaps, it often snaps at the point where it attaches onto the body. So a butterfly or a bird or anything that has wings, where the wings attach onto the body, the center of whatever we're talking about, it often needs to be reinforced. You often need like quite strong connections or structures in place to support where the wing attaches at the top, so up here, and where it attaches at the bottom. So we get essentially greater compression, probably at the bottom, and tension forces, like trying to pull it apart, and here trying to squash it down as the leaf sort of folds down, or the wings fold down. But we get essentially greater forces acting at the top here and at the bottom here. And so to help overcome that, basically we get some thickening of the cell wall. They're essentially just a bit tougher. Now underneath the vascular bundle here, there are some cells that are maybe still a little bit smaller or don't necessarily have that thickening, so they're just considered parenchyma again. So the main tissues you can probably split this into is your parenchyma, which is just most of the stuff that fills the leaf. So that's your mesophyll, plus maybe a little bit down here that doesn't have chloroplasts in it. Your collenchyma are a little bit more sort of structural support. Something like celery has a lot of collenchyma lining just under the epidermis to make it that rigid, tough stick-like structure. So here we have a little bit at that point where it's going to have the most stress. And they've done experiments where if they sort of shake plants to stimulate wind movement, they find a lot more collenchyma, or rather that the cell walls of those collenchyma cells are even thicker, maybe twice as thick, due to that mechanical disruption or that added stress. So your mesophyll layer, the palisade and the spongy mesophyll, are part of the parenchyma. A little bit more parenchyma down here. Collenchyma, bit of structural support added at the bottom here and at the top. Epidermis is technically part of the parenchyma. It's often referred to as specialized parenchyma. Your xylem and phloem can be considered different tissues. They have a different function. And then the bundle sheath cells, often around the xylem and phloem, are often considered a sclerenchyma. Now, I wouldn't really expect you necessarily to know all of those, at least not at this stage. When we cover transport in plants, we'll look at that sort of stuff in a lot more detail. But I guess the key thing here is if you can't distinguish between the palisade and the spongy mesophyll, you can call them just a mesophyll layer, or you can even just refer to them as parenchyma. In fact, if you can't make out something, parenchyma might be a good guess as to what's in here. If there is a little bit of a neck at the top here, around sort of connecting the circle, which is the vascular bundle or the vein, to the top, that's probably collenchyma. And down the bottom here, especially if you see cells that have quite a dark outline, and just under the epidermis here, these ones at the bottom look to me like they might be slightly darker around the edges, so maybe we've got a bit of collenchyma there too. So that's really those tissues. I hope that makes things a little bit clearer. But again, I wouldn't stress too much about that, especially for the prac assessment. And at this stage, you'd probably be asked to label a few things, like 2, 3, maybe 4 cell types, layers, structures on this picture. And I think there's plenty of things there you could do: the epidermis, the palisade, the spongy, the vein. That would be it already, or xylem, phloem. There you go. You've already got a few more. So I think there's no shortage of things you can actually label. Now I've got a few more here just to show you that the arrangement can be a little bit different sometimes. So here we've got an epidermis on both sides. I can actually see on the bottom a little bit of a cuticle here too. So I might be thinking that, this might be the top, but actually I've definitely got my palisade on this side, so I'm pretty sure this side here is still the top. Although I see some stomata here, some pores, so it seems a bit odd to have the stomata on the top. So I'm not actually 100% sure which is the top and the bottom on this one. It's not actually too important. I think from the shape, I'm still fairly certain it is the right way up. So I'd be thinking this is my palisade mesophyll, epidermis at the top, epidermis at the bottom, probably spongy mesophyll down here. We've got a vein here, bundle sheath cells around the outside. Xylem would be these big ones, phloem the smaller ones. This bit here, you might be wondering what the hell's going over there. That's actually just a vein that's going sideways. So it's one of these that's been chopped, essentially looking at it sideways. So in here we would have maybe our xylem at the top and the phloem at the bottom if it's arranged like it is here. It's just a lateral vein, one that's running rather than going towards or away from us. It's running sideways like this. Okay, so I think there's more than enough things we could identify there. These would be our stomata. We can see a little airspace there going in there, and the guard cells there. Cool, here's another one. This one's actually already labeled for us, so that helps. I can see the epidermis on both sides again. Palisade mesophyll, spongy mesophyll. This is one thing that students often do badly. They assume or they draw the palisade mesophyll continuing all the way across the top. Look at the top here in the middle. It's not the same as the palisade mesophyll either side. So this is a little bit of my collenchyma. And that's there just again, like I said, to provide the structural support. Spongy mesophyll just underneath, we can see that either side. Again, that doesn't carry on all the way across. We've got that central vein kind of blocking the way there. Alright, so xylem, the large ones in here, phloem, some bundle sheath cells around the outside, or that sclerenchyma. Down the bottom here, possibly still a little bit more parenchyma, and then the last ones here that are sort of a bit bigger with sort of darker outlines are probably collenchyma again. Alright, here's another one, so a little bit different. We've got the epidermis either side, it's stained quite differently, so that's quite nice. Can't really quite make out the palisade and spongy. I think the palisade's actually on this side, on the lower side, so maybe we're looking at things upside down. But these look a little bit more sort of rod-shaped or regularly arranged to me, and on this side looks like there's a little bit more sort of air spaces. But I'm not 100% sure, so on this one maybe I'd just label this mesophyll rather than separate into palisade and spongy. And we can see clearly though the xylem and the phloem, so xylem here, phloem here, and the bundle sheath around the outside, and possibly a little bit of collenchyma, maybe parenchyma, but probably collenchyma at the top and the bottom, or it could even, it looks like they're quite similar to the bundle sheath cells. Again, if you're not 100% sure, stick with the ones that you can definitely label. I can see here a stomata, alright, some guard cells, definitely here, even some on the other side too. Sometimes we get them on both sides. Tend to get more on the underside of the leaf, but like I said, often both sides. Here's another vein, Alright, this is probably the central one, this is probably the middle of the leaf, even though the leaf doesn't have that typical sort of fold or V-shape. Here's another one, so bundle sheath cells, some xylem, some phloem. Yeah, essentially here, there's more than enough I think we can label, and it's still a pretty straightforward diagram. Going on to the next one here, okay, same sort of things. Here we can see a nice, I put this in one in because you can nicely see the cuticles, probably this red layer here, and you can see it's not cellular. We can't make out any cells there, so that is a secretion. The epidermis directly underneath, on either side. H here, there we've got some guard cells, or the stomata, it looks pretty closed at the moment. We've got the bundle sheath cells, which make up the outside, so this is the vein, E. We've got xylem, D here, phloem is F. Here the slightly smaller ones. Again, possibly some collenchyma at the top and the bottom here. Not 100% sure on that, but probably. And again, we can see another vein over here. So if you were drawing these, like, maybe draw 2 of them, maybe don't have to draw in the third one here. Draw 2 or 3. You wouldn't have to draw 7 if there were maybe 7 of them. Your diagram is supposed to be representative or show some of that structure. So draw a couple to show there's not just one in the middle, but you wouldn't have to draw a whole bunch of them. I mean, hopefully you'd want to draw your diagram, I guess, big enough that you're never going to see 20 of them at the same time. Also, you don't have to go right to the edge, draw right up to the edge, so you can leave a little bit of a gap. Unlike your cell diagrams, you're never going to be able to draw the entire structure kind of finished. I mean, the leaf is usually too wide for that. Okay, here's one more. This one seems a little bit more messy. And if you get something like this, I mean, this would be an unfamiliar structure to you. You'd be looking at this going, oh, what's this? If you got that structure and if it looked unfamiliar, I mean, just draw it, but don't label it. You've got to draw what you see there, but if you're not sure what you're looking at, label the things that you are sure that you're looking at. So here we've got the epidermis. I'm sure you could all pick that out. On the lower surface we've got a bit of an epidermis too. On this side it's not, on this last little bit here it's not so clear, but I would assume it's still there. I mean, it's here, it's along the top. We're going to assume there isn't just a random patch where it's missing epidermis. Underneath, this is probably collenchyma, this layer here. Palisade mesophyll, spongy mesophyll, where there's all this air spaces. Alright, we've got our veins here, so bundle sheath cells, xylem and phloem. Can't really necessarily see xylem and phloem separately. I'm guessing the xylem are at the top there. I can just make out some little circles, and at the bottom I can't at all, so that's probably phloem. But again, if you can't really make it out, draw the outside, label the whole thing a vein. And if it's getting too small to differentiate the parts, between the xylem and phloem, then don't, and just draw the whole thing as the vein. You can probably just make it out, so xylem here, phloem here, maybe here it's a little bit clearer, xylem, phloem, probably just under there, sort of more a smile shape. Now this here, okay, we've got the epidermis continues around. Alright, you can see definitely on this side maybe, the epidermis continues around, so it comes in a loop. And then there's these little points or spikes, they're actually like little hairs that come off from the inner surface there. And then we've got, these are actually stomata. Now they're probably not clear enough or big enough to draw. I've actually got a little video of me drawing this particular slide in one of the other tutorials. And so I drew the epidermis coming around like this. I drew the little hairs. I ignored the stomata, they weren't clear enough for me, so I just left them out of my drawing. These are actually called stomatal crypts. It's a little indentation, so it helps prevent the water loss. Gas exchange still occurs, but because they're not sort of exposed so much directly to the light, and these hairs try to sort of help trap moisture too. So they're designed to allow for gas exchange and again minimize water loss. But again, it's not a structure you'd need to be familiar with, and if it was in there, or at least not at this stage, and so if it was in there, you draw it in, and you just wouldn't label it, label the things that you are familiar with. So possibly down here, a bit of collenchyma again, bit of reinforcement, not 100% sure, once again, could still just be parenchyma. Can be a little bit difficult to tell. I mean, we're looking for a difference in the thickening, how thick the cell wall is. Not so apparent. So again, I'd just stick with the things I know. I'd label a vein, possibly xylem phloem, epidermis, palisade spongy mesophyll, and that'd probably be enough. Alright, that's it.